Assalamu alaikum students, myself is Bushra Khan and I am from Eam Grammar School and College, and today I am going to teach you mechanism of enzyme action and specificity of enzymes. It is present on page 1111 and ends on 113 of your textbook. Learning outlines include, first of all, mechanism of enzyme action, which includes its procedure. Then we have lock and key model, and in the last we discussed induce feed model. The second heading is specificity of enzymes. The subtopics comes number of known enzymes, then we have examples, and the last one is determination of specificity of enzymes, means how you determine the enzyme specificity. First of all, we have mechanism of enzyme action. Dear students, kindly open your book and take out page 1111 and understand the procedure. Mechanism of enzyme action. When enzyme attaches with substrate, a temporary enzyme substrate complex is formed. Enzyme catalyzes the reaction and substrate is transformed or changed into product. After it, the ES complex breaks and enzyme and product are released. Now come to equation. First of all, we have take enzyme molecule, which is represented by E, and substrate molecule, which is represented by S. By the combination of both, we get ES complex. It is a temporary phase, and after some time, substrate converts into product and enzyme molecule released separately. This is the procedure of enzyme action. Now come to lock and key model. Lock and key model. In order to explain the mechanism of action enzyme, a German chemist, Emil Fischer, in 1894 proposed lock and key model. According to this model, both enzymes and substrate possesses specific shape that fit exactly into one another. This model explains enzyme specificity too. Then we have induce fit model. In American biologist Daniel Koshland in 1858 suggested a modification to lock and key model and supposed that induced fit model is best model. According to this model, active site is not rigid. Active site of any enzyme is not rigid, rather it is molded into required shape. Molded means changeable. It can be changeable into the required shape to perform its function. Induced fit model is more acceptable than lock and key model of enzyme action. Now come to specificity of enzymes. We have number of known enzymes include 2000 known enzymes are present, each of which is involved in which one specific chemical reaction occurs. Enzymes are also substrate specific. Examples include the enzyme protease, which breaks peptide bonds in proteins, will not work on starch. Means the enzyme of protein, that is actually protease, does not work on starch molecule or lipid molecules, and lipids have other molecule and starch have different one. The enzyme which work on starch is actually amylase, and the lipase enzyme works on lipids and digests them into fatty acids and glycerol. These are the examples of enzyme specificity. Then we have how we determine the enzyme specificity. Specificity of different enzymes is different or determined by the shape of their active sites. Means the active sites present in enzyme molecules determine the specificity of enzyme. Active sites possess specific geometric shapes that fit with specific substrate. Now come to the book pages. Mechanism of enzyme action is shown. First of all, we have discussed the equation of enzyme action and the whole procedure. Then we have the first paragraph, that is actually lock and key model. Then we have induced fit model, and we have discussed specificity of enzymes, in which known number of enzymes are discussed. Then examples are present here, and then how we determine specificity of enzymes. Every substrate has its specific shape and active site that suits that substrate, fits that substrate into it. These are the related links. Thank you.